Hello and welcome to this session of videos where I'm going to prepare for clearing out a few of the critical concepts of the few chapters of biology for the entrance examination of NEET, which is an entrance examination people opt in India to go to medical school and medical colleges. So I hope that these videos will meet up to your expectation. I have never done videos before. This is probably the first time I'm making it, and I'm really worried that how it will turn out. And please do let me know if you're able to understand from these videos, or if you are having any problems regarding understanding, or if you are really struggling with any concept what I should talk about in my further videos. Okay, so today's video, if we talk about today's video, we're gonna talk about the topic of microsporogenesis. And this topic has been discussed by many people in your school, in your coaching institutes, and so on and so forth. In today's discussion, what we're going to focus about is to make you guys aware of the boundary of this process. So if you are in class 12th, you probably by this time be aware of there is a chapter called sexual reproduction in flowering plants. And in this chapter, what we see is that we discuss the sexual reproduction of flowering plants in great details. And if we look at the process of sexual reproduction, we see that sexual reproduction has now been subdivided into three stages. And this we all know, this we all have studied in our first chapter, which is reproduction in organism. So there we see that sexual reproduction, this is subdivided into three stages, and these stages are: first one is pre-fertilization, second one is fertilization, and then the third one is post-fertilization. So microsporogenesis, this is the event of pre-fertilization. So pre-fertilization is a time when we see there are two major events are happening. The first event is gametogenesis, and the second event is gamete transfer. So these two sections are there in the pre-fertilization events. Now talking about flowering plants or angiosperms, these are like the most advanced category plants what we have in our planet. And in this type of plants, we see that they have a highly reduced gametophytic structures. Okay, so over here, what do we generally try to approach is basically a comparative study, you can say. And the comparison we will draw with the gymnosperm, because gymnosperm and angiosperms, these two set of plants are the only plants where we can see the seed production is there. So they are evolutionarily closely related to each other. We will also try to give some of the details about the pteridophytes, along with some of the comparison we'll draw with pteridophytes and the angiosperms. Okay, so the first thing first, what people are aware of, what you are aware of, is that flower, this structure is a it is a reproductive structure for the angiosperm. Okay, and we see that in flower we have four worlds. We have the world of calyx, we have the world of corolla, we have the world of androecium, and we have the world of gynoecium. So here you see that androecium and gynoecium, these two portions are the essential world. That means these worlds participate in sexual reproduction in a more active manner. where if we talk about calyx and corolla, they are known as non-essential whorl, and they do not participate in active sexual reproduction process, but it helps in the process of protection. It generates protection, and it also helps in the process of pollination. So that portion we will discuss in our gamete transfer part. Now, coming down to the section of the essential whorl, we have the essential whorl of androecium and gynoecium. So we will first discuss the androecium. So androecium, this is a whorl of stamens, okay, and stamens, these are the male reproductive structures, okay. So over here you see that, you know, a typical stamen, it is having a filament and it is having an anther portion, okay. So to begin with, filament and anther, both of these two structure are sporophytic structure or diploid structure, okay. Now you see that in the portion of anther, there is the presence of two lobes. Two lobes are there, and these two lobes are present in most of the anthers. So that's why we say that anthers are typically bilobed. In each lobe, there is the presence of two sporangia. Okay, so together it will become 4 sporangia per each anther. Hence, we generally say that typical anthers, they are also tetrasporangiate in nature. Okay, so anther, this portion is the fertile part, and filament portion is the sterile part. Anther is present at the distal end of the stamen, so that you need to understand. Now, if we look at the anther, we see that anther is basically made up of two portions. It has a anther wall portion, and then it has a portion of sporogenous tissue. So sporogenous tissue, they are present. They're present almost at the center of each sporangia, and the anther walls and the sporangia walls, they are synonymous. Okay, so do not confuse yourself with these two terms of anther walls and sporangia walls. They are basically same. Now, if we talk about the anther wall and sporangia wall first, and then we'll talk about the let's say the sporogenous tissue. We first will begin that discussion with a little bit of ontogenetic history. So ontogenetic history or developmental history for anther is basically talks about a situation of how the anther walls and sporogenous tissue have developed within the anther simultaneously. Now, if you look at it, you see that in the process of development, there was the presence of a cell, and that cell is known as archisporial cell. Now, this cell was gone under transverse division, and after the transverse division, you see it has given rise to two cells. A cell which is present on the above side, it is now known as parietal cell. The cell which is present below, it is now known as sporogenous cell. Now this parietal cell, it will go under mitotic divisions, and it gives rise to the anther walls or the sporangia walls. On the other hand, the sporogenous cell, it goes under the mitotic divisions, and it gives rise to a sporogenous tissue. Okay, so first we will now discuss the anther walls. Okay, so anther walls, these are basically prepared for two purposes. There is a purpose what we call as the purpose of protection, and the second purpose is to give nutrition. The third purpose is to give dehiscence of pollen grains. Now, if you see that if we talk about the anther walls, we have 4 set of anther walls. Okay, so we have a epidermis, then we have a endothecium, then we have a middle layer, and then we have a tapetum, the innermost layer. Okay, so if we discuss that, okay, we have to understand one thing, that from outer side to inner side, okay, this chronology has been maintained. So from outer side to inner side, the sequence is our epidermis, then endothecium, then middle layer, and then tapetum. Okay, so first outer three layer, if we talk about, okay, the epidermis, endothecium, and middle layers, these three layers of anther, they perform two common functions. Okay, and these are like very frequently asked question. So the common functions what they perform is the protection and the dehiscence. So if there is a question asked about the fact that what are the common functions of the three layers, outermost three layers of anther, so the answer will be protection and dehiscence. Okay, so now if we talk about tapetum, the innermost layer, that is solely nutritive in nature, and we'll talk about like how they will give nutrition and what kind of functions they will perform. Now to start with, we'll first talk about the outermost layer of anther. And if we talk about the outermost layer of anther, we see that our outermost layer is epidermis, and epidermis is mostly it is mostly single layered, and it is having a function of protection. And there are only few plants where we see that this layer, this epidermis layer, can also be fibrous in nature. And if it is fibrous in nature, then we call that particular type of epidermis as exothecium. Okay, and this type of examples we see in RC euthobium and different types of insectivorous plants, etc. The next layer we'll talk about is the endothecium layer. Okay, this is also a single layer. Okay, and if we talk about endothecium, we see that the endothecium cells are having alpha cellulosic thickenings in their cell wall. So it is present as a band in their cell wall, and these alpha cellulosic thickenings are present and it gives an edge to this layer, that this layer is hygroscopic in nature. So it is moisture sensitive, it can absorb the moisture from the environment. But having said that, if we look at hydrophytes, okay, in hydrophytes, they are already living in water, so they do not require extra moisture. Okay, so hence in hydrophytes we see that these alpha cellulosic thickenings or these bands are absent. Now we will talk about the portion of middle layers. Okay, so middle layers are these are multi-layered. It depends upon, this can be 3 layer, 4 layer, up to 6 layer, depending upon the families of angiosperm we are discussing. Now middle layers, these are very interesting because they do not present in mature anther. They degenerates in mature anther. They are present at the young anthers. Okay, so these particular layers, these are having a nutritive property, so they degenerates over the period of time to give nutrition to the developing anther. Now if we talk about the innermost layer or the tapetum layer, so again tapetum layer is a single layer, it is the innermost layer, and also this is it's a layer where the cells will degenerate over the period of time. Now the difference is tapetum degenerates and tapetum also gives nutrition, but it gives nutrition to the developing pollen grains. Whereas if you talk about middle layers gives nutrition to the developing anther. Okay, so this is the like clear sort differences what we have. Now, if we talk about tapetum, we see that tapetum are of two types. We have granular tapetum or secretory tapetum, and we have amoeboidal tapetum. So the difference is that in case of amoeboidal tapetum, we see that the cells of the tapetum, it will burst open, and it gives the nutrition to the developing pollen grain, and it's mostly a primitive class of tapetum. If we talk about the secretory or granular tapetum, we see that the cells, they slowly secrete out the products in the pollen grains, and they go under, like it slowly goes under degeneration. Okay, so beside that, there is one thing which is very important about tapetum cells, is that tapetum cells are polyploid in nature. So they are polyploid, and they're also at times they're just rich with DNA. So why we are saying these things? Because the DNA enrichment in our tapetum cells, it can happen in various ways. Okay, to start with, if you find that a cell get stuck, okay, if the cell gets stuck at S phase, so then you know what happens in S phase of the cell. The cell, it participates in DNA replication, and there you see that the number of chromatids, it's keep on increasing, but still all the chromatids, they're joined with the same point, and which is your centromere, correct? So over here exactly same thing can happen with these cells, because where we see that these cells can also go under uh, repetitive DNA replication. They're stuck at S phase, and they are keep on increasing their chromatid number and the DNA amount, but the chromosome number is still fixed. Okay, so this set of strategy to enrich DNA, this is known as endo-reduplication. Now, it can happen that the cell may get the cell may have participated in the process of DNA replication, and then after that it has entered the mitotic phase, and then you find that the chromosome separation didn't occur well, and you find that now the cell is having it has increased the number of chromosomes. It has almost doubled the number of chromosomes. So this is known as endomitosis. Next, we'll talk about there are situations where we see that the chromosomes have segregated to each poles, and after that, irrespective of forming two nuclear membrane individually, there is only one nuclear membrane what has formed by fault, and that has bring both set of chromosomes together. So this type of nucleus which is present in the cell is known as restitution nuclei. There can be cytokinase failure as well, which can lead to the multinucleated condition in tapetum cells. So all these properties basically gives rise to a condition where the DNA has enriched in tapetum cells. Tapetum cells, they also important for the production of various materials which are very important further for the process of sexual reproduction. So tapetum cells, they produce compatibility proteins, they produce sporopollenin, they produce pro-Ubish bodies, they produce Ubish bodies. They are important for the production of pollen kit and etc. Callose sugar, they produce callase enzyme. So all this material they produce, and these all materials take part in the process of sexual reproduction. Now this is all about the anther walls. Okay, so next we'll come down to the sporogenous tissue. Now, this sporogenous tissue, this is a portion where we see that the sporogenous cell, from the archisporial cell, it is now going under repetitive mitotic divisions, and then it has developed as a tissue. Now this tissue, initially they are highly vascularized tissue. They are connected to each other by the help of the plasmodesmatal connections, and after that you see that once the anther is mature, the process of microsporogenesis will start. When that time comes, you see that few of these cells from the sporogenous tissue, first they block these plasmodesmatal connections, okay, and they isolate themselves by the help of callose sugar. So they first deposit callose as a plug to this plasmodesmatal connection, and then they dissociate themselves from the tissue, and now they become an individual unit. And this unit or these cells are now having a callose coating on top of it. Okay, so these cells are now known as microspore mother cells or the pollen mother cell. One has to keep in mind that whatever we have discussed so far, except tapetum is polyploid, except tapetum everything is diploid. Okay, so now this pollen mother cell, what we have seen that it has been now isolated and it has prepared itself, it will now go under the sporic meiosis. So it will go under the sporic meiosis and it will give rise to 4 cells, but these 4 cells will be stick together because the callose coat outside of it is still intact. So this give rise to the condition of pollen tetrad. Okay, so the process of production of the microspore mother cell to pollen tetrad, this is where the meiosis ends. Okay, after that you see the callase enzyme will work on the pollen tetrad. It will dissociate the callose coating and it will release these haploid cells from the pollen tetrad, and each of these haploid cells are now known as microspores. Okay, so this process, this is the boundary, okay, from the pollen mother cell or the microspore mother cell to the production of microspore. This event only is known as microsporogenesis. Okay, now what we need to discuss right now over here, which probably will going to give us a better understanding, is a comparative study. And this comparative study is with the pteridophyte and with the gymnosperm. Okay, so if we look at pteridophyte, we see that pteridophytes, they had a leaf, and that leaf was a reproductive leaf. We call it sporophyll. And on the sporophyll, on the if we talk about the abaxial surface of the sporophyll, majority of the cases we have the presence of sporangia. Okay, and that sporangia carried the spore mother cell, which further led to the formation of spore. Now that was a homosporous condition for a pteridophyte. This you have studied in your plant kingdom chapter. We see that heterospory has developed. Heterospory has been seen in few of the pteridophytes and majorly seen in our gymnosperms, so and angiosperms. But now if you look at it, you will see that, look at the line of evolution. Imagine the line of evolution. So there was a leaf, okay, and the leaf was carrying the sporangia. So now if you look at like gymnosperm is also having the same thing. Gymnosperm is also having the sporophylls, but now they have 2 types of sporophyll. They have microsporophyll, where they carry the microspores, and they have megasporophylls, where they carry the megaspores. So one is for the male reproductive leaf, one is the female reproductive leaf. So now if you look at the angiosperm, as we have discussed, you see, look at the anther and look at the structure of stamen. So if you look at it, you will see like it is almost a super specialized leaf. Anther is a super specialized leaf. This is carrying the sporangia. So what we see as a sporophyll in pteridophyte and in gymnosperm, in angiosperm, we look at it as as stamen. The same thing we see, that the thing what we see as megasporophyll in gymnosperm, it further convert or modify into the form of carpel, the female reproductive structure in flower. The last thing what I want to touch over here and discuss is that in both of the cases of pteridophyte as well as gymnosperm, we have seen that the reproductive leaf, they can form a cluster, and these clusters are known as strobili or cone. So I would like to ask you a question, and also would like to ask you I think what do you think about flower as a evolutionary structure. So we see anther over there, we see the ovaries over there, okay, which are supposed to be the sporangia kind of forms. So one is there with the stamen, another one is there with the carpel. So these are your specialized or modified reproductive leaf, and now they are present in a cluster. So you should make one concept clear in your head, that in evolution you see that strobili, which is present in the pteridophytes and which you see in gymnosperms, are now modified into flower, and that's why we call flower as a super specialized leaf. Flower is known as a modified shoot, that we all know. It is also known as a super specialized leaf. So this is all about today. We can briefly discuss one more part, which is the pattern of cytokinesis in microsporogenesis during the meiosis. So we'll just discuss two portions. The first thing is, if you see during the time of sporic meiosis on pollen mother cell or microspore mother cell, that cytokinesis 1 is present, and after cytokinesis 1 the karyokinesis 2 or meiosis 2 is beginning, and then the cytokinesis 2 is happening. So this is basically a process which is sequential in nature. So sequentially the cytokinesis 1 is happening first, and then cytokinesis 2 is coming. So this type of pattern, this is known as sequential pattern, or we call it successive pattern, and this give rise to a pollen tetrad which is known as isobilateral type of pollen tetrad. These are advanced class of microsporogenesis, and these we see into monocots. Now if you see that during the microsporogenesis, cytokinesis 1 is absent, and without cytokinesis 1 the two nuclei which has produced after meiosis 1 is now participating. It has gone under interkinesis, and now it is participating into meiosis 2, and at the end of the meiosis 2 you see that cytokinesis 1 and cytokinesis 2 are happening at the same time. Okay, so if it happens at the same time, 1 and 2, it will give rise to 4 cells at the same time. Okay, and this process is known as your simultaneous process, and simultaneous process give rise to the structure of pollen tetrad, which are known as tetrahedral structure of pollen tetrad, and these are primitive type of pattern for microsporogenesis, and these are seen in dicots. So this is about our discussion of critical session of day 1. In next session we would like to talk about it in the female reproductive part. Until then, like, stay tuned, and I would say, like, stay home, be healthy. That's all. And Yeah, that's all. I'm really conscious about making videos. This is my first video, so I hope you all enjoy it.